All right, module 7. This is the last of our sort of taxon-specific modules, and we'll be talking about the Eupolypods too, that other major clade of Eupolypods, and covering 4 families. So we'll end with a bang, so to speak. So, quick reminder, we just talked about the Eupolypods 1, this group that usually has round sori and has many vascular bundles in the base of the petiole. This is that weird character that may sound not very useful, but it's actually useful. If you're willing to break off a leaf, you can almost always tell which of these 2 major clades you're dealing with. We, however, for this module, are going to be talking about Eupolypods 2. They usually have linear sori, and they have 2 vascular bundles in the base of the petiole. So here's what they look like in terms of a phylogeny. This major clade up here. Here's the Eupolypods 1 that we just covered. So about a quarter of extant ferns are in this Eupolypods 2 group. A big group of ferns, and there's about 11 families of them. Fortunately, not all 11 families are here in California, so we don't have quite as many to deal with. But one of them is the Cystopteridaceae, the fragile fern and oak ferns. There's others elsewhere, but here in California, we basically only have the fragile fern. This is a small, inconspicuous fern that grows typically in rocky places. That is easily mistaken for an immature version of basically any other fern. One way to recognize it, first of all, is to turn the leaf over, look, make sure you have some sori. If you do have sori, if they're in great shape and you can see that they have this weird indusium that arches over like a hood that's only attached on one side, then you already know that you have Cystopteris fragilis. You've got it to species right away. But even if you can't see that, if you see that it is fertile, so it's not just a little leaf of a wood fern or something, and you see that the veins go all the way to the edge of the leaf, then you can be pretty confident that you have Cystopteris. Cystopteris happens to be my main study organism, and so I'm going to go on a bit of a tangent to say that Cystopteris fragilis, as is often circumscribed, occurs everywhere in the world that's within those red circles up there. So basically, it occurs across the entire northern hemisphere, but also elsewhere in the world, and even including typically all of these other colors too. So these are species that are sometimes considered to be different species, but often are considered to be fragilis as well. So a crazy distribution. What's going on here? Some of these plants grow in the understories of deciduous forests in Eastern North America. Others grow on cliffs in Scandinavia. Some are like desert ferns that grow in cracks and crevices in Utah, whereas others are epiphytes in tropical rainforests. Some are super lacy, have really finely divided leaves, whereas others have leaves that are only 1 cell thick and grow along waterfalls. So a huge diversity of morphologies. Why do we call all of these things one thing? Well, a lot of it has to do with a process called polyploidy. And what polyploidy is genome duplication, and it often occurs alongside hybridization. And so what you can have happen is you can have one diploid species and a second diploid species. So I've drawn whatever, I've shamefully used angiosperms in this case, but 2 different species of flower, and they happen to have different genomes. And they hybridize and then double to form what's called an allopolyploid that has both genomes of each of its parents inside it. So it's got a doubling of its genome and a hybridization event. And what this can do is it can make a new lineage, a new species that is perhaps intermediate between its parents, but is itself its own distinct thing. And it can be quite hard to tell either from morphological data or from molecular data what has really happened here. And if we do this in Cystopteris, for example, all of these green lines are these sorts of hybridization events. And all of the blue lines are what are thought to be allopolyploid types of Cystopteris fragilis. And the diploids are these things that are connected by black lines. So there's actually only 2 diploids here. And so what you can see happening is it looks like there's been a huge number of different allopolyploids forming from a relatively small number of diploids. They all look kind of similar. And so it's not that everything in the Cystopteris fragilis complex is the same thing. That's variable. It's just a whole bunch of different things where the boundaries between them are hard for us to determine. And so a big systematics problem is figuring out, how many species are there in Cystopteris fragilis and how do we tell them apart? So a quick little sort of tangent about some of the evolutionary biology that underlies some of these tricky fern groups that we find to be so challenging. All right. Our second family in the Eupolypods too is the Blechnaceae, the deer fern and chainfern and their relatives. They have linear sori with the indusia opening towards the vein. You see that, you're in Blechnaceae. A lot of species in this family are dimorphic, like this beautiful Cystopteris, the deer fern. You can really see what we mean when we talk about different leaves, different fertile and sterile leaves. Here's a view of those linear sori showing the indusia that open towards the midvein. Classic feature. Then I'll introduce the Athyriaceae, the lady ferns. This is a huge family elsewhere in the world, but relatively small here. These are also big, typically large ferns of understory, so you might confuse them with wood ferns, except they don't have a round sori. Instead, their sori are hook-shaped. And they have, because they're in Eupolypods 2 instead of Eupolypods 1, if you break the leaf off at the bottom, you'll see that they have 2 vascular bundles instead of a whole bunch. So it's going to take a bit of work to get familiar with lady fern versus wood ferns, but they are quite distinct. When they do have their sori back to back, they always open away from the midvein instead of towards it. So this is a tropical species we don't need to worry about, but just in case you were ever confused about it maybe being Blechnaceae, in Blechnaceae, the indusia open towards the vein, in Athyriaceae, they open away from it. And the final family I think I'm going to talk about, is the Thelypteridaceae, the marsh ferns. They generally have round sori, so they're an exception to the rule for Eupolypods 2. However, they're quite easy to recognize usually because they're usually hairy with these short, colorless, pointy hairs. So if you see something with hairs like this, you're basically guaranteed to have Thelypteridaceae. And many of the species are pinnate pinnatifid. So one's pinnate and then pinnatifid, and they look like this, pinnate pinnatifid. And this is a family that's actually been a subject of research here at UC Berkeley. So Alan Smith and Susan Fawcett. Alan Smith is probably the world's authority on the Thelypteridaceae. Susan just finished her PhD on the family, and they have revised the genus level classification. And so we now have a revised understanding of the relationships and the classification for the California species. So we have, instead of Thelypteris nevadensis, we have an Amorapelta, and Thelypis purpurula is a plasmoneuron. Again, I want to emphasize that pinnate pinnatifid. So this leaf is pinnate to make this leaflet, and the leaflet itself is divided substantially, but not all the way. So it's pinnatifid. Look, also, you see all those little hairs. This is a classic Thelypteridaceae, the marsh fern family, Thelypteridaceae. Here are the iNaturalist observations for the entire Eupolypod 2 clade in California. And so you see a lot of people observe chain ferns and lady ferns and deer ferns. And here's the Cystopteris. But there's a few other things that are seen, including some families that I didn't mention. Here's one woodsia and the Woodsiaceae, and here's a spleenwort. I didn't talk about the Aspleniaceae. There's actually a few spleenworts and 2 woodsias. But most of the species that you're likely to encounter are in those 4 families I just mentioned.